Hello everyone, today I'll be presenting a research paper titled Efficient mRNA Delivery to Resting T Cells to Reverse HIV Latency, published in Nature Communications in 2025 by Paula Savall and colleagues. This study focuses on one of the biggest challenges in HIV research, which is finding a way to target the hidden reservoir of HIV that current treatments can't eliminate. And during this presentation, I'll explain the problem, the research we're trying to solve, how they approached it, and the major findings of the study, and also why these results are important for future HIV cure research. Before proceeding to analyze the study, it is necessary to explain why HIV cannot be easily cured. First of all, it should be noted that the virus infects CD4 plus T cells, and at the moment, the antiretroviral therapy is highly efficient for managing the disease and suppressing its progression. However, it cannot be used for eradicating the virus completely. And the matter is that some infected CD4 T cells are in a resting, also known as latent stage. During this stage, the virus is not visible for both the immune system or the therapy. As you can see in the diagram on the left, HIV first infects CD4 plus T cells, then ART suppresses viral replication, but the latent virus remains hidden inside resting cells. If the treatment is stopped, those cells can become active again and begin producing the virus. Because of this hidden reservoir, one of the biggest goals in HIV research is finding a way to reach these resting T cells. And that's exactly the problem this study is trying to solve. Now that we've seen why HIV is so difficult to cure, the next question is, what were the researchers trying to accomplish? Their main objective was to determine whether a newly developed lipid nanoparticle called LNPX could deliver mRNA into resting CD4 positive T cells and reactivate latent HIV. What the researchers expected was that LNPX performed better than previous delivery methods because resting T cells are normally very difficult to target. If LNPX could successfully deliver mRNA into these cells, it could become a promising platform for future HIV therapies and help move HIV cure research forward. Let's now consider how the researchers carried out their hypothesis. The study consisted of 4 major parts. In the first part, they developed and screened different lipid nanoparticles in order to find the most effective one, which was LNPX. Then they examined whether LNPX was able to deliver mRNAs into the resting CD4 positive T cells using reporter mRNAs. This way, they could determine the efficiency of nanoparticle delivery into the cells. After successful delivery, they delivered that mRNA into LNPX in order to examine whether it is capable of reactivating latent HIV in resting CD4 positive T cells from patients infected with HIV. And finally, in order to test the safety of the treatment, they measured cell viability, tested T cell activation, and delivery of CRISPR activation system by LNPX. This set of experiments helped the researchers to examine not only efficiency but also safety of LNPX as a possible platform for new generation HIV therapies. The first major finding of the study was that LNPX was able to efficiently deliver mRNA into resting CD4 positive T cells, which is normally one of the hardest cells to target. Looking at the figure on the left, the researchers created LNPX by modifying the original patisiran lipid nanoparticle. They changed some of the lipid components to improve how well it could carry mRNA into the resting immune cells. Then, if we look at the graph on the right, we can see that as the dose of LNPX increased, the amount of mRNA delivered into the resting T cells also increased. That tells us that the new nanoparticle was working and as intended and became more effective at higher doses. This is an important result because one of the biggest challenges in HIV research is getting treatments into resting CD4 positive T cells where the virus can remain hidden. By successfully delivering mRNAs into T cells, LNPX provides a promising platform for developing future HIV therapies. Now that the researchers showed that LNPX could successfully deliver mRNA into resting T cells, the next question was whether it could actually reactivate latent HIV. To test this, they loaded LNPX with TAT mRNA, which is a viral protein that can turn HIV gene expression back on. If we look at the graph on the left, we can see that as the dose of TAT LNPX increased, the amount of HIV reactivation also increased. In contrast, the control treatment showed almost no reactivation. This tells us that the effect was caused by the TAT mRNA delivered by LNPX. And the graph on the right looks at cell viability and how healthy the cells remain after the treatment. And even after receiving the TAT LNPX, most of the cells stayed alive, showing that the treatment caused very little toxicity. And together, these results shows that LNPX can successfully deliver functional therapeutic mRNA that reactivates latent HIV while also keeping the cell healthy, which makes it a promising strategy for future HIV cure research. For the final experiment, the researchers wanted to see whether LNPX could deliver something even more complex than mRNA. So instead of that mRNA, they loaded it with CRISPR activation components, which are used to switch specific genes on without changing the DNA itself. The diagram on the left shows how this system works. The CRISPR activation complex is guided to a specific gene where it turns that gene on and increases its expression. Looking at the graph on the right, we can see that as the dose of CRISPR activation LNPX increased, the expression of the target gene CD25 also increased. The control treatment stayed close to baseline, showing that the increase was specifically caused by the CRISPR activation system delivered by LNPX. This is an important finding because it shows that LNPX is not limited to delivering TAT mRNA. It can also transport more advanced gene editing tools, and which makes it a flexible platform that could be used for many different HIV gene therapies. To wrap up, this study showed that LNPX is an effective platform for delivering therapeutic genetic material into resting CD4 positive T cells, which has been one of the biggest challenges in HIV cure research. Throughout the study, the researchers demonstrated that LNPX could efficiently deliver mRNA and successfully reactivate latent HIV using that mRNA and even transport more advanced tools like CRISPR activation components and also maintaining high cell viability. And although this is not a cure for HIV, these findings represent an important step forward because they provide a more effective way to deliver gene-based therapies into the cells where HIV remains hidden. And looking ahead, this technology could support future shock and kill strategies, improve the delivery of mRNA and CRISPR-based therapies, and help move this type of research closer to preclinical and eventually clinical studies. I think this study is a great example of how advances in drug delivery can help overcome one of the biggest obstacles in HIV cure research.